Hi friends, have you ever wondered how scientists can predict the genetic makeup of a particular population using maths? Yes, it is possible with the help of Hardy-Weinberg law. This Hardy-Weinberg law is an important topic in anthropology optional in UPSC, and it comes in paper 1 unit 9.3. In this particular session, we are going to learn all about it. So let us begin. As the name suggests, Hardy-Weinberg law, it is formulated independently by two individuals. One is Hardy, who is a British mathematician, and another one is Weinberg, who is a German physician. So what does it describe? Well, it describes the relationship between the frequencies of alleles and genotypes in a population under certain ideal conditions. So the principle, it states that the genetic variations, that is the variations in the alleles and genotypes in a population, will remain constant from one generation to another generation if there is no evolutionary influences, that is under the absence of variations. If there is no variations, then there won't be any change in the genetic composition. The genetic composition is going to be constant, and that is what they are calling it as genetic equilibrium. Now, since it is a mathematical and the genetic in synthesis of both of them, we need to look at the mathematical derivation of it on the basis of the genetic principles that we have earlier learned. So before going into the derivation of the equation, let us look at what are the assumptions, because we are talking about a ideal condition which does not exist in the reality. It is a hypothetical situation. So what are those assumptions which create these ideal conditions? Let us look at them. Well, the first assumption is the population size should be very large. It is so large that the genetic drift, that is the random fluctuations that happen in the allele frequencies, are negligible. See, in the earlier sessions, I have explained this. The genetic drift is directly proportional to or inversely proportional to, directly proportional to 1 divided by population size drift is directly proportional to 1 divided by population size, or you can say it is inversely proportional to population size. Lesser the population size, more will be the impact of the genetic drift. If the population size is large, then the impact of genetic drift, which is a random fluctuation, it does not affect much. That is the reason why first assumption is the population should be very large. The second assumption is the mating should be random. Such a randomly mating population, we call it as pan-mectic population. So the population we are considering is a pan-mectic population where each and every individual is a potential mate and the mating is completely random. There is no preference for the specific genotype or a trait. That is the second assumption. The third assumption is there is no mutation. That is, there is no random inheritable change in the genetic material. So that is the third assumption. And the fourth assumption is there is no migration. Like if there is a migration, then there will be a gene flow. So again, there will be variations. So that is the reason why there is no migration, and thereby there is no introduction of the new alleles. And the final assumption is no natural selection. See, natural selection also brings certain changes. When it is absent, then what does it mean? All the genotypes which are present in a particular population, they have equal fitness. None of them are more fit and none of them are less fit. So less fits are discarded by the natural selection and more fits are selected by it. So such selection is not there. So under these assumptions, we get an ideal condition, And if such ideal conditions are there, from one generation to, let's say, another generation, what is happening? The genetic composition is being constant. This is the condition which they call it as genetic equilibrium or Hardy-Weinberg equilibrium. Now, let us try to derive it and learn it from the mathematical perspective. For this, let us take a small example. That is, let us take one character or one trait with two alleles. Let's take, which is a very well-known example, that is the height. We have already seen in Mendelian experiments, one of the trait that has been taken is the height of the plant. Now, if you take the height, you can have two alleles. One is tall, another one is the dwarf variety. Now, out of which one of them is a dominant one. Let me write it as capital T. Another one is the recessive one. Let me write it as small t. Now, let us take the frequency of the dominant allele is P and the frequency of recessive allele is Q. Then we know that at any case, the sum of the frequencies of all the alleles must be equal to, that is P plus Q must be equal to 1. So that is what we are writing it here. Now this is with respect to one allele. One allele. So the frequencies of this should be equal to 1. If you wanted to consider the entire genotype which is having two alleles, then it will be P plus Q into P plus Q. This is nothing but 1 into 1. So on the other side we get P plus Q into P plus Q is equal to 1. Simply multiplication of it. So if you multiply P into P plus P into Q plus Q into P plus Q into Q is equal to 1. So here P into P means it is a homozygous condition for a dominant allele. So we can write it as P square. Since we have taken the frequency of the dominant allele is P, so you can write it as P square plus here PQ or QP. Both of them are heterozygous conditions for a dominant allele. So we can write it as 2PQ and Q into Q. This is homozygous condition for recessive allele. So here the recessive one is having a frequency of Q. So you can write it as Q square. So this is equal to 1. This is what we call it as Hardy-Weinberg equation, right? So here, to summarize this, what is the meaning of it? The frequency of dominant allele in homozygous condition. Okay, let me write it as homozygous dominant allele plus the frequency of heterozygous dominant allele plus the frequency of homozygous recessive allele, all of them are equal to 1. This is what is your Hardy-Weinberg equation. So let us make the genotypic table for it in order to understand it in a better manner. Say we have already considered T, capital T is the dominant allele, small t is the dwarf allele. So now when we cross these two individuals with these characteristics, so what are the possibilities that we can get? Capital T, which is homozygous dominant allele, and another one capital T, and small t, capital T, both of them are heterozygous. Okay, let me write heterozygous, and this is again dominant allele. Say capital T is present, and finally we'll get small t. This is homozygous recessive allele. Now if we add all of them, this is nothing but t into t plus capital T small t plus small t capital T, which you can write it as 2 into capital T small t plus small t into small t. Say this is the addition of all this. So what is the addition of all of it? It is again coming to be 1, because these are all the alleles. The sum of all the alleles, the frequencies of all the alleles here, it is going to be what? It is going to be 1. So that is how we can derive it, and we can say this is the Hardy-Weinberg equation. Now it has a lot of applications, because it's a simply like in mathematics, if you don't know something, what we do, we assume that it is present. Then we start doing the derivation and we come to a contradiction where we will say our assumption in the initial case is wrong and this cannot happen in the contradictory manner. So that is what here they are doing. First, they are assuming that there are ideal conditions, and based on those ideal conditions, they are deriving it, and they supposed to get the entire value to be equal to 1. But in reality, it won't be 1. So that is when they say there is a deviation from the equilibrium. That means there is no ideal condition. That means there is genetic variations. Using that, we can understand what is the impact of natural selection or mutation, how much genetic load is there, how much deviation has happened. So it has a lot of applications. Let us look at some of them. First, see, it helped in assessing genetic drift. What is the meaning of drift? Drift is the random change. So whenever there is a random change in the gene composition, obviously there will be a deviation from the expected value of the Hardy-Weinberg equation. So the equation is not going to be equal to 1. So whatever the deviation that has happened from the 1, that can be due to genetic drift. That is how we can calculate the magnitude and the presence of the drift. This can help us in knowing the random change that has happened in the frequency of alleles. Similarly, it can also be used to detect the role of natural selection. Right, again here we will be calculating the genotypic frequencies using Hardy-Weinberg expected value or equilibrium, and if there is any deviation, it will suggest us that if there is any departation from here, it would suggest that there is a natural selection working on the genotypes. Similarly, we can also measure the role of the migration. So how much variations or how many new alleles have been introduced due to migration can be calculated using the Weinberg equation. All of them based on the contradiction. Like you calculate, given the frequencies, you calculate the expected outcome. It's supposed to be 1, but if there is any deviation from the 1, that will give us the hint that certain things have happened, right? Similarly, identifying carrier of recessive disorders. See, in human genetics and medical genetics, the Hardy-Weinberg equilibrium, it can be applied to estimate the frequency of carriers like the heterozygotes for recessive disorders. See, if there is a homozygous recessive one, they are already affected. But there can be heterozygous recessive ones where the dominant gene is is expressing and it is not letting the recessive deleterious genes to express. So they will be having these recessive genes as the carriers. So we can understand this particular frequency as well. Then one more application is conservation of genetics. That means, say, deviation from the equilibrium. It indicates what is the risk or what is the genetic load in a particular population genetics. Based on that, we can make like crucial changes, and this can help us in making informed conservative decisions with respect to the genetics. Then it also helps in the forensic genetics. See here, the equilibrium is used to estimate all the frequencies in a population, which is important for calculating the probability of matching DNA profiles. See here we use DNA profiling to identify criminals, victims, missing persons, etc. There it has applications. Similarly, in plant and animal breeding, like we can get hybrid varieties, the variety of plants and animals with the desired characteristics. That can be done here. So this helps in developing breeding strategies. Similarly, for studying evolutionary changes, see, research study the evolution of the populations using this equilibrium, then the amount of deportation of the genetic material that has happened at successive levels can tell us what is the trend in the evolution. That can be understood here. So these are all some of the applications. Now, to conclude, it is a valuable tool for understanding the genetic dynamics of populations and can be applied across various fields, which can be used in inheritance patterns, genetic variations, understanding evolution, and even for making predictions about genetic frequencies in the future. So these are all the no applications and no scope of Hardy-Weinberg equilibrium. I hope it is clear. If it is, just hit the like button and share it with your fellow aspirants so that it can reach to the maximum number of aspirants. And also do tell me in the comment section what is the next topic that you want me to make a special video so that I can decode and explain it in a simple terms. See, there were so many previous year questions from this particular section. You can take up any particular question and you can submit to the given mail ID. Thank you very much, everyone. See you in the next video with another topic. Till then, take care. Bye, everyone.